Genetic resource banks, or what we call gene banks, are a type of a biorepository which preserves genetic material, and it is a collection of seed plants, tissue cultures for potentially useful species, especially those that contains genes of significance to the breeding of crops, and this is important for food security. In plants, this could be done by freezing cuts from the plants or stocking the seeds of those plants. With corals that we find in the aquatic biodiversity, fragments are taken from them and they are stored underwater tanks under controlled conditions. So corals are the marine invertebrates that form compact colonies of many identical individual polyps. And usually these coral species include important reef builders that inhabit tropical oceans and secrete calcium carbonate that forms hard skeleton. Plant genetic material in a gene bank is preserved at -196 degrees Celsius in liquid nitrogen as mature seed dry or tissue meristems. In plants, it is possible to unfreeze the material and propagate it. However, in animals, a living female is required for artificial insemination. In animals, this is done through freezing of sperms and eggs in zoological freezers. However, it is difficult to utilize frozen animals and sperms and eggs, although we have been able to do it successfully, but it is not so successful as compared to plant material. The database of the largest gene banks in the world can be queried via a common website called Genesis, which is the web interface for being able to access gene banks in the world. So the history of genetic resource banks started way in the 1882s, where Alphonse de Cadell was the first botanist to attempt to explain how to locate the origin of plants, and he published a book of origin of cultivated plants. In 1905, we had the conservation of plant genetic resources being initiated in India. In 1926, Vivelov, a Russian explorer, geneticist, agronomist, organized a worldwide exploration for collection of seeds and propagating material of large number of cultivated plants, wild and even related species. In 1951, Vivelov again proposed about 8 centers of origin and 3 sub-centers for different plant species. In 1961, FAO organized the first international technical meeting on plant exploration and introductions. You can see most of the history began with plants before we could advance to animals. In 1968, Sir Otto coined the term genetic resources as we use it today to create awareness to plant breeders of the gradual loss of germplasm. Then 1968 also, we had the Crop Ecology and Genetic Resource Unit of FAO being established after their meeting in 1961. In the 1970s, USA initiated action to collect, conserve, evaluate, and utilize plant germplasm resources when there was the southern corn leaf blight disease outbreak. So they were sensitized that they need to conserve to ensure that they preserve this plant for future. In 1974, International Board of Plant Genetic Resources (IBPGR) was established in Rome. In 1976, a National Board of Plant Genetic Resource was established in New Delhi, again for crop species. And then in 1985, we had a National Board of Animal Genetic Resource was established in Kamal for animal species. So you can see actively this is when we had resources for animals. And then 1992, the IBPGR that was made here in the 1974 was transformed to a new autonomous organization called the International Plant Genetic Resource Institute, IPGRI, to assist developing countries in the conservation of genetic plant and animal resources. What is the purpose of gene banks? Of course, it's a physical facility for maintaining collections of live plant materials, domesticated cultivated plants, wild plant species, crop wild relatives and other wild plant species useful for food and agriculture, and of course other end uses. We will do or store entire plants, seeds, pollen, embryos, meristem, cells or DNA, depending on what is the biology of the species. So it's a facility for maintaining crop diversity, and when we have now animal, it will maintain animal biodiversity. Usually this diversity is in the form of seeds, and they're usually stored in frozen state. Some gene banks use normal household freezers for this purpose. When, for example, you're not freezing for a long time, the ideal temperature is between -10 and -20. Each different type is stored in its own container, such as a bottle, and sealed with aluminium foil for packaging. Gene banks, of course, exist to conserve genetic diversity of wild and domesticated organisms that humans will depend on for food, fiber, medicine, and energy. Over 7.2 million plant germplasm accessions are housed in approximately 1,750 national and international gene banks. So you can see we have quite a good number. Therefore, to conserve agricultural biodiversity, gene banks are used to store and conserve the plant genetic resources of major crop plants and their, of course, crop wild relatives. There are many gene banks all over the world, with Svalbard Global Seed Vault being probably the most famous one. So we've been talking about accession, here. So accessions, it's a number or it's a common term given to an individual sample in a gene bank, such as a distinct species or variety. So it's kind of an identity number. Gene banks manage both genetic resources and information about those resources. Diverse genetic resources are essential in improving the productivity of nutritional quality and sustainability of agricultural systems. Rapid scientific and technological developments have changed the way scientists explore and understand natural variation. So gene bank managers must stay abreast, of course, with scientific developments to fulfill the responsibilities to the public, because we keep having new breeds of both plants and animals, so they need to keep updating their gene bank information. So we have various types of gene banks: seed bank, tissue bank, cryobank, pollen bank, field gene bank, sperm bank, and ova bank. So a seed bank, just from the word, it preserves dried seeds by storing them under very low temperatures. Spores and pteridophytes are conserved in seed banks because they can be dried. But other seedless plants, of course, such as tuber crops, cannot be preserved in this way. So these ones we'll see they are usually preserved in form of tissue or tissue banks. The largest seed bank in the world is the Millennium Seed Bank, housed at the Wellcome Trust Millennium Building in London. Now tissue bank now will of course encompass every other species of crops that cannot be put in form of seeds. So they are going to use buds, protocombs, meristematic cells, and they are conserved through particular light and temperature arrangements in a nutrient media. And of course, this helps us preserve seedless plants and those that reproduce asexually. Cryobank is from the word cryopreservation, so it's a technique where seed or even embryos are preserved at very low temperatures, usually minus 96 degrees, that is in liquid nitrogen. And it's very important for helping conserve species facing extinction, because at this temperature you can store for 1,000 years, more than 1,000, thousands of years. Pollen bank, this is a method of storing pollen grains. Of course, you know these are useful in reproduction. We can make plants which are facing extinction in the present while using this technique. So again, you can enhance or facilitate reproduction or breeding of plants using pollen stored in pollen banks. Field bank, from the word field, is a method where you plant plants for conservation of genes in a specialized ecosystem that you have constructed artificially. So you create a segregated field within a facility, and then you maintain growth of certain plants over a period of time, or even as long as the plants are still facing challenges of extinction out there. Through this method, one can compare even the difference among plants of different species in detail. This is also useful in evaluation. We shall be seeing that throughout the gene banks, we need to carry out what we call evaluation. After we have stored these seeds, these tissue meristems, we need to keep ensuring that they're actually viable. So a field is also one way in which we can plant the seeds, plant the meristems, and still see if the germplasms that have been stored are still viable. A sperm bank is where now we have semen facility that collects and stores human sperm donors for, of course, solving infertility issues. So these are called donor sperms, and the process, of course, of now using them is through artificial insemination or IVF. Ova is now the other way. Now you store the egg, which you call the donor eggs. And of course, this is also for infertility issues, to assist women who are not able to produce ova, and help them through artificial insemination or in vitro fertilization to be able to conceive. So in a gene bank, usually we have certain activities that occur, and the main activities in the development and management of a gene bank will include: the first step, of course, is to collect and acquire what is your germplasm, what is your genetic material, what is it a seed, is it an embryo, is it a meristem, is it a tissue? And then, of course, process. The second step is processing, assessing the quantity, how much, how is it viable, what is the health of the sample, and then prepare it for storage, depending on if you are doing extreme freezing or freezing at lower temperatures. Then, of course, we have storage. You have prepared it for storage, now store in a cold store, laboratory, or even in the field like we have seen. Then, of course, we have regeneration and multiplication. Periodically, you determine the amount of time or what are the intervals of trying to rejuvenate and increase the material, so that with time you are sure that it is viable. So you get out a certain accession number of germplasm, test it in the field, rejuvenate, and produce even more, and then store again, collect, process, store. Okay, then we have characterization and evaluation. We need to constantly do characterization of our if it is a seed bank, we need to keep checking what are the maybe seeds that we are having, what are their sizes, what are their species, if you plant them in a field, how 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 do they grow, how many days do they require, how often do they, after how long do they produce flowers, after how long can they produce other seeds, etc. And then of course we do documentation. A lot of documentation and inventory must be maintained and made available for each of the samples. And then distribution, sometimes or most times we store these things in the gene bank because there will be consumers or requesters of the same, so we need to distribute clean, disease-free seeds or other planting material or even animal material for propagation and breeding. So I have put some components and activities of a community gene bank illustration. If we have this gene bank, you can see that we have various storage of various specimens. Some of them are dried seeds, others will be certain tissues. So in this case, we are going to have, of course, the seed bank and the field bank. Seed bank because of the storage of the seeds, and then field bank because we said we need also to have a land, an artificial ecosystem where we grow. So some of the activities that will occur in such a situation, of course, will be, there will be storage space. It will provide an operational manual, linkage and collaboration with even other maybe sub-centers and national gene banks, red listing of extinction, endangered species, among others. We talked about regeneration and multiplication of what already exists. We're able to multiply. If it is seeds and they are not in good numbers in the ecosystem, we can be able to regenerate and multiply. Of course, characterization and evaluation, genetic enhancement, we can be able to adjust genes so that we make or improve breeds, collections and storage. We have diversity fair. Yes, we can have fairs and shows within certain community gene banks for people to go and see what are the diverse species within their community. Seed processing, even in readiness for planting. A learning center for children, young people, old people, traditional knowledge of what exists within certain communities. It's a form of an agricultural tool center. And then what we call a seed herbarium or museum, where we are going to have a display of the various types of species that we have. A seed herbarium is a catalogued collection of plant seeds using identification and research on plants, and you can use it to create a museum within a community, which even international collaborators can come and see. Of course, you connect it to a database even for more wide coverage across the world. It forms an image bank for people to visualize the various species across the community. And of course, a diversity block because it contains a variety of plant species, known and unknown, new and upcoming, among others. So that is generally how a gene bank is important within a community. And of course, this also preserves biodiversity for future generations. So another very important thing is how we also merge in situ and ex situ, because germplasm and gene banks are ex situ. We are storing out of the natural environment. So for example, if we are talking about a farm will have cows, sheep, goats, horses, pigs, chicken. So that's on farm population of different given species. Then we can be able to do gene bank collections, okay, so that once we have done gene bank collections of these different species, we can be able to, after we have collected and stored within gene banks, so we can be able to restore certain species, what we call regeneration and multiplication. and then we can be able to reintroduce lost variants, okay, within the in situ conservation to increase variability. We can also be able to update collections now from the in situ. We can be able to update these collections and provide an insurance for future generations. And then of course we have other ways in which we're able to analyze the gene bank collection using genetic techniques or molecular techniques such as single nucleotide polymorphisms. The differences between these species can be studied. You see, we're able to compare species 1, 2, 3, 4, 5, 6 using SNP array, and once we do that, we can be able to even give the differences between species over time, and that way also be able to generate what we call new breeds, because once we understand the genetic makeup and differences between the organisms, we can even engineer or do genetic manipulations and improvements. So now I have kind of mentioned gene databases. So what are gene databases? So we've been looking at gene resources or gene bank resources or genetic resources. So now we need this information to go far and wide. The information that you could be having in a community database concerning maybe crops or plants that exist, plant species that exist within a certain local area, need to be accessible to the national conservation authorities, and beyond the national, we also need the international community to access. And this now only happens when we use what we call softwares or computer programs that can help us to access and retrieve information concerning genetic resources across the world. So a genetic database is one or more sets of genetic data, can be in form of genes, gene products, variants, phenotypes that have been stored together within a software to enable us to retrieve genetic data. Also add, once we keep discovering and do research, we need to add in the information in these databases, as well as extract information from the data during research. If you're a scientist, then you need to research. Genetic databases are one of the richest sources of genetic information. So I give some few examples of gene bases that exist in the wild. We have what we call Gene Bank. It's a resource of the National Center for Biotechnology Information, NCBI, that usually will centralize gene-related information into individual records. They actually also use accession numbers, so you can be able to get the genetic sequence of most of the organisms in the world. And then we have what we call a genome database, GDB, a public repository of the data on human. Now this is specific for human genomes, clones, STCs, polymorphisms, and maps. So for the human genome, you're going to have also genetic markers and molecular markers that will exist in that repository. We have others like for crops, the International Multi-Crop databases, like the Crop World Relative Global Portal, SINGA, PGR Forum, GeneSys, which is very important for accessing most online databases, EU plant variety databases, among others. Then we also have what we call the CGIAR Gene Bank platform, and some of the partners in Kenya here will be the IRRI, the International Livestock Research Institute in Kenya, where you can, especially as biotechnologists, you can even do internships and work in such an institute. We have the National Gene Bank now here in Kenya, the National Gene Bank of Kenya, GBK, that was established in 1988 by the Kenyan government, is a national institution for coordination and implementation of all activities concerned with crop, plant, and forage genetic resources. And then one that I would want us to look at is what we call the Genetic Resources Research Institute of Kenya, Agricultural Livestock Research Organization. which what we call Gary Calro, which is important now for animal. You can see that this is for plant and this is for animal genetic resources. So simply, a genetic database is a software where we find all genetic information of all germplasms that have been stored within the various national, subnational, and even community gene banks. So I want us to do one of these, the web-based database that is Gary Calro. So this is the website for Gary. We are saying we are using Genesis as the program to access this database. So this database, the Genetic Resource Research Institute of Kenya Agricultural and Livestock Research Organization is responsible for conserving plant, animal, aquatic, and microbial genetic resources. And the mission is to harness, conserve, and promote greater sustainable use of genetic resources for increased, of course, food security, resilience of agricultural production systems, as well as improved health and socioeconomics. So currently, this institute has over 50,000 accessions belonging to about 1,800 crop species of mainly land races, improved cultivars, okay, that you can that is sold to farmers for cultivation, and of course their wild relatives acquired through some of them through donations and also field collection from various parts of Kenya. So far, about 23,000 accessions have been characterized, and characterizations means we know what are their species, we understand their growth, we understand their genomic sequences, we understand which areas they grow in, among others, to determine, of course, what's the diversity and how variable are they. Characterization data plays critical role in understanding the value of our germplasm and facilitates access and utilization already. So we are saying characterization helps us to understand the value because we know this is these characteristics, this is how valuable it is, where does it grow, etc. So if you look at this website, of course, it will give you a lot of data. So I want us to just focus on the recent data sets. We have so much that you can get from this website. So I will just pick on one. Maybe let's use one that is close to Kirinyaga. Characterization of maize accessions. So all the maize that have been submitted to this Gary Caro from Embu. So let's look at morphological evaluation of maize accessions conserved at Gary to assess the diversity of the accessions. So you can see how many have been donated or collected, about 179. And the number of traits, this has already been characterized. So these 179 have about 19 traits. So the rest of the traits are similar. Then this was evaluated in the 1992. Of course, it gives us the metadata. Remember, we talked about a lot of recording of information. Who collected, who is the curator, who are the managers, and where do they come from. Then the data set use and licensing. It has been licensed. Then the data and resources. So the traits that have been observed: days to 90% maturity, number of days from sowing to 90% of plants reaching physiological maturity. Also, we talked about 19 traits. Then national accession number, of course, is given. Seedling vigor, visual measurement recording 3 weeks after the emergence of the seedling. What's the plant height? What is the ear height? Days to 50% flowering, midrib color, lodging, number of ears per plant, basal tillers, ear length, kernel rows, ears characterized, number of ears randomly harvested, kernel uniformity, endosperm color, kernel type. kernel color, endosperm texture, 1000 grain weight. You can see all that information. Those are the various characteristics that have been analyzed. Then accessions, the list of the accessions included in this data set. So you can see all these are the accessions. We said they are about 179, so there are quite many. So maybe we can just pick one line. Let's pick this one. Yes, so this is GBK 032993. Of course, the genus is Z, the specific or species is maize. So Z maize, specific name Z maize Erva H511 line 225. It's a maize crop. And then, of course, found in Kenya. Then the location of collection is Embu. Then passport data completeness index, genus 120, subtaxa, all those details concerning that particular plant. And you can see we are able to access as much information. And when was it lastly? We talked about updating this information. You can see that it was first entered in 1992, but it was last updated on 13th of July 2018. So you can see, so in short, what happens in these databases, you can be able to get as much information as you need depending on what you want to find out. You can be able to get the information concerning the particular plant species and where it comes from and what are the various characteristics of the same. So unless there is a question, that is it on gene banks.